The secret itch pathway. The brain must be able to distinguish between gentle touch, which is necessary for many activities, and mechanical input, which causes an itch and necessitates scratching. The brain must be able to distinguish between gentle touch, which is necessary for many activities, and mechanical input, which causes itching and necessitates scratching. These nerve fibers carry the impulse to the dorsal horn of the spinal cord, from where it spreads to the thalamus and other parts of the brain via cells that have the gastrin-releasing peptide receptor, GRPR. Sensory neurons transmit light touch information from the skin to Y1 neurons in the spinal cord, as seen in a dorsal root ganglion cross-section. Mechanical itch is caused by low-threshold mechanoreceptors, LTMRs, in the skin. According to a previous work by the same group, inhibitory interneurons, INs, in the dorsal horn of the spinal cord that express the chemical neuropeptide Y-CRE control impulses from these cells, NPY-CRE INs. Mice without these INs suffer chronic scratching for no apparent cause, as well as scratching in response to minor mechanical stimulation. Despite the ablation of the GRPR neurons, this remained constant, indicating that mechanical itch is communicated independently of the GRPR expressing neuron route. As a result, the NPY-CRE INs regulate how mechanical itch sensations are transmitted from the spinal cord to the brain. According to a new study, these peptide signaling neurons process mechanical itch and deliver the signal to specific dorsal horn neurons that contain the proteins Y1 and LBX1, Y1-CRE neurons. When Y1-CRE neurons are destroyed in NPY-CRE ablated mice, chronic scratching is removed. In normal mice, ablation of Y1-CRE neurons reduced itching in response to mechanical stimulation. Drugs improved the scratching response to a mechanical itch and triggered spontaneous scratching in Y1-CRE neurons. That is, the Y1-CRE neurons in the dorsal horn form an excitatory circuit that is necessary for mechanical itch to appear. The researchers observed that the body's NPY is essential for Y1-CRE cells in the dorsal horn to stop signaling. It does this by reducing responses to light touch, such as that produced by a hair, and mechanical itching. Mice with the LBX1-CRE/Y1F/F conditional deletion scratch and react excessively to itch, causing mechanical stimuli due to a lack of endogenous NPY, but not chemical itch and pain. This shows that NPY-Y1 signals in the dorsal horn of the spine regulate mechanical itch impulses. Activating NPY-CRE reduces mechanical itch and sensitivity to light punctate touch by reducing Y1+ cells in the dorsal horn. Although many cells express Y1-CRE, just a few are required for mechanical itch response. Mechanical and chemical itch have independent pathways from the receptors to the dorsal horn of the spinal cord, based on the lack of effect when these neurons were destroyed. Modulating LTMR receptor-mediated feelings and separating mild touch from irritating sensations requires the NPY-Y1 pathway. Because of the presence of this gated channel for mechanical itch, humans can benefit from the benefits of light touch.
Thank you for watching. Hope you will like the video I created for you. Please support me, click on subscribe. Then click on the bell. Please tap the below to view videos of the same series.